So I come from the lab of Center for Functional Genomics and Biochips, which is part of Faculty of Medicine and University of Ljubljana. And we use omics technologies and system biology tools in liver cancer, and we are interested in studying mechanisms, biomarkers, and therapeutics. And maybe I will just show a bit what we do on the projects that have been published. Here is one of those projects. We work a lot on circular RNA and all other non-coding RNA, which have molecular mechanisms in HCC, and we studied also expression in patients. So we have expertise in all circular RNA and other non-coding RNA related experimental approaches, and we can also measure these RNAs in patient samples, and also, for example, here is a ddPCR in plasma of patients, and we collaborate. There are only 2 surgical teams in Slovenia who operate these patients, and I have active collaborations with them. And we also have cooperate with a company to develop ddPCR biomarker panels. Another approach that we do a lot is use, for example, genome scale metabolic models, functional genomics, and also machine learning applications to study transcriptome studies and transcriptome data. And here is some publications about liver fibrosis in animal models. And here we collaborate with Faculty of Computer and Information Science with Computer Structures and System Laboratory, who do all the genome scale metabolic and mathematical modeling, and also some kind of combination in artificial intelligence. And then this is just the project that started and is funded by our national agency, where we plan to sequence using nanopores, so long-read sequencing, to poly-A and circular RNA sequencing in 3 different samples from HCC patients who want to do differential expression, discovery of novel circular RNA, alternative splicing, and then we want to use systems biology, machine learning, and functional genomics tools to identify potential circular RNAs that we will validate then in patient samples for biomarker potential, and in vitro systems for carcinogenic and also therapeutic potential, which is something that we want to work maybe further as RNA-based therapeutics in liver cancer. And here we have, again, collaboration with both clinical centers, and there is also in Slovenia only 1 liver onco gastroenterology unit, which we also collaborate and get samples. So, and here we collaborate with another team at the Faculty of Computer Information Science, the Bioinformatics Laboratory. So here is all the things that I could remember that we have at our institute and our faculty. So we cover different omics platforms, the long read, the short read, single cell, targeted metabolomics, nanostring, proteomics. And for all of these, we offer experimental and data analysis support, which is usually more important for people who come from the laboratory. And we also, since I have active collaboration with clinical partners, you can also get samples or fresh samples to do tumor organoids. But we have also animal facility laboratory for extracellular vesicles and all sorts of multimode readers, imager packs, and microscopes for live cells imaging. And furthermore, I'm part of Elixir system biology group, where I have several collaborations in this group, and where you can find maybe the content that would be interesting to you for your project. And at the end, I also have a supercomputer to offer additional support. So I have, so we do a lot of training in data analysis. We do every year a symposium. We don't only work, we also celebrate parties. We go for trips. And I worked the last 3 years as evaluator of this MSCA project. So I have I exactly understand what our national contact point talked about. And also our university, and as you have seen, our ministry have full support in preparation of the proposal. And that's it. Thank you for your time.